Hello everyone. I welcome you all to Biology Lessons with Kalyani. Today we will be discussing about the protein structure, which is the part 2 in the lecture session about proteins. So let's look into the topic. As we have discussed in last lecture session, proteins are basically polypeptides and they are made up of different amino acids which are linked up by peptide bonds. Now proteins exhibit 4 levels of structure. Okay, the first level in this is primary structure. Now primary structure of proteins refers to the unique sequence of amino acids present in the polypeptide chain. Right? Now this unique sequence of amino acids is determined by the genes. Now what happens? DNA contains the genes and in the process of transcription they lead to the formation of mRNA. And during translation the linear sequence of amino acid is formed or the polypeptide chain is formed, that is referred to as the primary structure of protein. Clear? Now, what does this primary structure of protein give detail about? This primary structure of protein tells about the positional information about the amino acid. What does this mean? It says what are the types of amino acid present in the polypeptide chain and what is the sequence of those amino acids. Right? Now this primary structure is responsible for what is the type of protein formed or specificity of a protein. Right. Now it also helps us in determining the physical and chemical properties of the protein. So these are the things which the primary structure of the protein tells us. Now this primary structure of protein is very important because if there is a change in single amino acid, that will lead to the abnormality in the protein structure. And we have many genetic diseases and these are due to the abnormality in the primary structure of protein. Right? So see, this is the polypeptide chain. Different amino acids are joined together. In the extreme left, we have the amino group, which is also called as the N-terminus. And in the right, we have the carboxyl group, which is called as the C-terminus. Right? Now, what happens? The linear structure of protein that is formed after the translation, it doesn't remain like that forever. That extended rigid rod-like structure of polypeptide, it undergoes different type of twisting or folding, and that is referred to be as secondary structure, which is the second level of protein structure. Okay, we have two main types of secondary structure, alpha helix and beta sheet. Indian scientist Ramachandran made a significant contribution in understanding the spatial arrangement of polypeptide chain. So Ramachandran sir is very important in this context. You'll be reading more about it in the higher classes. One thing you must note here is that if you consider a polypeptide chain, the type of twisting or folding which is present in one portion might be different from the other portion. What does that mean? It means that some of the portion of the protein might have an alpha helix, some might have a beta type of conformation. Okay, so let's look into what is alpha helix and beta sheet in detail. Now, the most common type of secondary structure formed by proteins is alpha helix. Here what happens? The polypeptide chain, it forms a spiral structure. Okay. Now, this structure was proposed by Pauling and Corey, and here the Alpha helix is tightly packed coil structure and amino acid side chain extend outwards from the central axis. And this structure is stabilized by extensive hydrogen bonding. Now all the peptide bonds except the first and the last in a polypeptide chain participate in hydrogen bonding. Let's now look into the structure of alpha helix. So this is how a polypeptide chain twists and form a spiral structure, and this type of structure is called as alpha helix. Now for this you have to remember that proteins always exist in the right-handed alpha helix. Now what is right-handed alpha helix? Look, this is your left hand, this is your right hand. If you twist the protein towards the right side, it is called as a right-handed helix, and if you twist it towards left, it is called as a left-handed helix. So in our case, proteins take up this right-handed alpha helical conformation. Now I told this alpha helical structure, it is stabilized by the hydrogen bond. Look, this is your carbonyl carbon and this is your oxygen and this is your NH. So hydrogen bond can be formed between the H and O. So these type of many hydrogen bonds are present which stabilize the alpha helical structure. And where are the side chains present? The amino acid side chains are always present towards the outside of the alpha helical structure. Okay. Then there is a term called as pitch. In the helix, we define pitch as the distance between any two turns. So this is one turn, this is another turn. The distance between the two turns is called as pitch. Now it also can be defined as the vertical distance for one complete turn. Means if you consider this point and when it takes a 360-degree turn or reaches the same point, the distance traveled vertically is referred to be as pitch. Now, what are the amino acids? How many amino acids can be formed in this pitch of the alpha helix? There are about some 3.6 amino acids which will be present inside one complete turn of alpha helix. Now, pitch is 5.4 angstrom in case of alpha helix. What does that mean? It means that if you take a complete turn and you measure the vertical distance, it is 5.4 angstrom or 0.54 nanometer. Now, distance between each amino acid, how can we calculate that? It is equal to pitch or the vertical distance taken up by the complete turn divided by the number of amino acid in that complete turn. That is equal to 5.4 divided by 3.6 because we have 3.6 amino acids in a complete turn. So that gives 1.5 angstrom. So if you take two amino acids, the distance between the both is 1.5 angstrom in case of an alpha helix. Another point which you have to remember here is that if you consider the hydrogen bond, we already discussed that the first and the last amino acid, they do not take part in the hydrogen bond. Also, hydrogen bond takes place between every fourth amino acid. Every fourth amino acid participates in the hydrogen bond in case of your alpha helix. That's all about the structure of alpha helix. Now you can just remember some examples of it. Alpha keratin, which is present in hairs and nails, is an example of alpha helix. Now, let's see the second type of secondary structure, that is beta pleated sheet. Now this beta pleated sheet type of structure is very common in the silk fibrin protein. This structure was also proposed by Pauling and Corey, and in this case what happens, two or more segments of fully extended peptide chains, they form hydrogen bonds between them and they make a pleat type of structure. Okay, what happens? You have a polypeptide chain. Let's see, you have a polypeptide chain here, and this is the segment. These different segments of a polypeptide chain will come close like this, and they will form the hydrogen bond. So that will give a pleat type of structure, and such secondary structure is called as beta pleated sheet. Now, if we consider the directionality, beta pleated sheet type structure can be of two types. It can be parallel type of beta sheet, or else it can be anti-parallel beta sheet. The polypeptide chains in the beta sheet may be arranged in the same direction, making the parallel beta sheet, or anti-parallel, making the in which the chains are in opposite direction. Now we can have the beta pleated sheet between two different polypeptide chains if the protein is made up of different subunits, or a single polypeptide chain can fold back on it forth on itself to make a beta pleated sheet. So let's look into the structure in a little more detail now. Let's now look into the structure of beta pleated sheet in a little more detail. Now this is the primary structure of the protein. So what happens if the polypeptide forms a zigzag way of folding? So what happens? This segment forms the bond, hydrogen bond with this segment. So the directionality of the protein of the polypeptide is not changing. So this type of beta pleated structure is known as parallel form. So if you just consider this section, these two sections, so what happens? The amino group and the carbonyl group, they arrange like this, and two segments are stacked up in this way. Okay, now the carbonyl oxygen and the hydrogen of the amino group, they form a hydrogen bond. If you just notice, then the hydrogen bond is at an angle here. Okay, now see this zigzag line I have marked in yellow dashed line. Okay, so that this structure becomes clear. So what happens if you look from the top? It gives an appearance like this, like lines stacked up, zigzag lines stacked up upon each other. Okay, now if you consider another way, which is called as your anti-parallel beta pleated sheet, here what happens? Polypeptide, instead of making a zigzag line, it forms a loop here. See, so if you like pinch a part of polypeptide here, just like a loop, what happens? The directionality changes. So instead of N and C as we are having in the parallel, here it changes to N. It is making a N and C, it is making the bond with anti-parallel type of the structure. So look, this type of structure is called as the anti-parallel beta pleated sheet. Now, how does the hydrogen bond take place here? If you see, this is the bond between the oxygen and the hydrogen, and this is hydrogen bond is perpendicular. So, anti-parallel beta pleated sheet structure is more favored in case of the protein secondary structure. I hope the beta pleated sheet type of structure is clear. Now this is the overview. Like if you see from the top, this is how the anti-parallel type of structure looks, and this is how the parallel type of the structure looks. This is the three-dimensional view of the anti-parallel and parallel type of beta pleated sheet structure. Look, the hydrogen bonds are perpendicular here, and they are at an angle in the parallel sheet formation. So that's all about beta pleated sheet. Let's move on to the tertiary structure. Now, tertiary structure, in addition to the long protein chain, it is also folded upon itself like a hollow woolen ball, giving rise to tertiary structure. So here we saw in primary we just have the peptide bond. In secondary, along with peptide, we are having hydrogen bond, right? Now in tertiary structure, in addition to the hydrogen and the peptide bond, we will have many other bonds such as disulfide bonds, ionic interaction, that is the electrostatic bond, hydrophobic interaction, and van der Waals forces. So what happens? These type of interactions make the protein even more folded. Now the protein looks as if it is folded upon itself like a hollow woolen ball, and this type of structure is referred to be as the tertiary structure. Now this type of structure is responsible for the biological activities of the protein. So proteins, they attain the functionality or functional biological activity only in the tertiary structure, right? Primary structure gives the specificity of a protein, and tertiary structure is responsible for the functionality of the protein. Very, very important. So if you consider enzymes, which are nothing but proteins, so all the enzymes, all the proteinaceous enzymes, they have a tertiary structure, right? Because they are going to catalyze the reaction, so they will have a tertiary structures. Also the immunoglobulins, they will also have a tertiary structure. Now tertiary structure is basically the three-dimensional view of a protein. Now we have something called as domains in the tertiary structure. They are basically the functional unit of the protein structure. Now let's look into what are oligomeric protein. So until now we discussed the proteins which have a single polypeptide chain, but there can be proteins which have more than one polypeptide chain. So look, this is just one polypeptide chain. If more polypeptide chains are present in a protein, that type of protein is called as oligomeric protein. Now, in case of oligomeric proteins, the individual protein subunits, they are called as either monomer or protomer or subunit. So, if we say a dimer protein, that means that it has two subunits. If we tell it is a tetramer type of protein, it has 4 subunits. Now why I am talking about oligomeric protein? Why? Because if we talk about the fourth level of protein structure, that is a quaternary structure, that is exhibited by the multi-subunit protein. So let's look into more detail about it. Quaternary structure of the protein. Some proteins which are oligomeric proteins that contain more than one polypeptide chain are an assembly of more than one polypeptide chain or subunits, and the way in the which the individual folded polypeptide or subunits are arranged with respect to each other is called as the quaternary structure of the protein. Is that clear? Look, there will be one polypeptide. It will have a primary structure. It will folded or twisted after that to make a secondary structure. Then it will have a tertiary structure. Now these polypeptides having tertiary structure will again interact with each other to form a quaternary structure of the protein. Now in which way these individual folded polypeptides or subunits are like interacting with each other, that is called as the quaternary structure of the protein. So they can be linear string of spheres, they can be spheres arranged one upon each other in the form of a cube or in the form of plate. That is the architecture of the protein, that is referred to be as quaternary structure of the protein, right? So quaternary structure is shown only when the protein are oligomeric in nature or they have more than one polypeptide chain. So in this case, what are the type of interactions that play? So the interactions can be hydrogen bond, hydrophobic interaction, as well as the ionic bond. One of the example of quaternary structure is adult hemoglobin. Now this contains 4 subunits. 2 of the units are identical to each other. So adult hemoglobin is having 2 alpha subunits and 2 beta subunits. Total there will be 4 subunits. So look, this is your adult hemoglobin. It will have 2 alpha and 2 beta, and it will possess your 4-degree structure or quaternary structure. Very important example. Let's now see what are the types of bonds which help in stabilizing the protein structure. Now, protein structure can be stabilized by covalent as well as a non-covalent bond. So, look, what are the covalent bonds that stabilize the structure of protein? You have peptide bond which is present in the your like main amino acid chain. Apart from that, you have a disulfide bond, that is an example of the covalent bond. So here disulfide bonds will be definitely present between amino acids which are sulfur in them. That example is cysteine. So two cysteines, they will make bond with each other to form cysteine. So that is present in your tertiary type of and quaternary type of protein structure, right? So apart from that, you can have non-covalent bonds which also help in stabilizing the protein structure. Examples of it can be hydrogen bond. We saw already carbonyl oxygen and H of the amino group that were forming hydrogen bond in secondary structure, both alpha helix in beta pleated structure. Okay, another example can be hydrophobic bond, in which the hydrophobic amino acid like isoleucine and leucine form a hydrophobic interaction. Third example of non-covalent bond can be formed between charged amino acid. So we have aspartic acid which is negatively charged, lysine which is positively charged. So they can interact with each other and can form the ionic bond. So where do we find the hydrophobic bond and the ionic bond? We will be finding it in the tertiary type of structure. Also quaternary may also show the hydrophobic bond and the ionic bond. So these are the types of covalent bond we already discussed: peptide bond and disulfide bond. And non-covalent bonds can be hydrogen bond, electrostatic bond. Hydrogen bond is formed between nitrogen and the carbonyl oxygen. Electrostatic bond between the positively and negatively charged amino acid. And you can also have hydrophobic bonds which is formed between the non-polar side chains or non-polar amino acid, and generally you get the hydrophobic bond when the protein is in the aqueous environment. Apart from that, we have a fourth type of bond, that is the van der Waals forces. If that is a non-covalent association between electrically neutral molecule, and you can have permanent or induced dipole interaction which can help in forming the tertiary structure of the protein, right? So remember the bonds in primary structure of proteins, you just have the peptide bond. Secondary structure, peptide plus hydrogen bond. Tertiary structure, apart from peptide hydrogen, you can have disulfide bond, you can have ionic bond, you can have your electrostatic bond between the charged amino acid, hydrophobic bond, and van der Waals forces in both tertiary and quaternary, right? I hope you are now clear with the different structural details about the 4 levels of protein structure. I would highly recommend you to check out the part 1 of the proteins where we discuss the general overview. If you like this video, please like, share and subscribe to my channel. And if you have any doubts, please feel free to comment below. Thank you everyone. Have a very good day.